Hello, everyone, and welcome to our experimental design notes video for pages 2 and 3 of your notes. So in this video, we're talking about why do scientists collect data? We're going to go into how would scientists communicate data, how we measure data, and the parts of a graph. So let's go ahead and get started with why do scientists collect data? The first reason that we collect data is to have a written record. It wouldn't make sense for us to design an experiment and test it, but not write anything down. Then we wouldn't know what the relationship is. So we need to have it written down for us to look at. We collect data because it gives us a place to observe patterns. We need to collect the data, look at everything that we find, and we might be able to see patterns in that. And that means that there might be a relationship between the things that we're testing. We collect data in order to draw conclusions about the patterns that we're seeing and the relationships that we're finding. We also collect data for evidence. That means that we collected this data, we communicate that to other scientists. It's evidence that this relationship exists or doesn't exist. One of the main reasons that scientists collect data is to answer a question. Any question we have about the real world, we could create an experiment for and try to answer. We collect data in order to communicate our ideas. Communication is one of the most important things that scientists do, because if we collect data and don't communicate it, then we did an experiment and didn't tell anybody, so nobody else can learn from that. When we communicate those ideas, we're able to make ideas more repeatable. Other scientists can try to test that and see if they get the same results. And when they get the same results and that idea becomes repeatable, we increase validity of that experiment. So we have more confidence in the data that we collected. So now we're going to get into the parts of kind of an experiment and what that looks like. So the first part is variables. Now, a variable is a quantity that is being measured or studied in a scientific investigation. And there are two types of variables. The first one is manipulated, or independent, as maybe you know it previously. The manipulated variable is what's being changed in the experiment. And then we have the responding variable, which is also known as the dependent variable. And that is a variable that changes in response to the manipulated variable. So it's fair for us to say that we can't have a responding variable without the manipulated variable. And without the manipulated variable, we wouldn't have responding variables. So here's a little example. Let's say we want to test how much water makes our size of the leaves, or that's what it looks like, size of the plant, number of leaves, if it's alive or dead. So the independent variable, the variable that's being changed, is the amount of water that we're giving the plant. The dependent variable, or our responding variable, is the variable that's affected by that change. So the size of the plant, the number of leaves, if the plant lived or died. So we could see that this medium amount of water made the size of the plant the best, provided the most leaves. The most amount of water caused the plant to die. So this amount of water affected these variables. So the next type of variable is a control variable. Now these are things in an experiment that stay the same. We also have the control group. And the control group is not changed, and that's for comparison purposes. But not all scientific investigations have a control group. So looking at this, they're changing the amount of liquid, or the type of liquid used to water each plant. So here we have pop, they're doing juice, and then they're doing water. And so our independent or manipulated variable is the type of liquid that we're using to water the plant. Our dependent variable, or the thing that's changing because of the liquid we're using, is how tall the plant is going to grow, or the health of the plant. So in order for these two things to happen, we had to keep some things the same. So constants, the type of plant that was used, these are the same types of plant. We also kept the pot size the same. A, B, and C are all the same pot size. They receive the same amount of each liquid, so maybe 500 milliliters of soda, 500 milliliters of juice, 500 milliliters of water. They kept the soil type the same, so the soil that's in each one of these is the exact same, and that just helps to eliminate any other possible relationships that might be happening. Now, being that we're providing this pot C with water, that would be our control group, because that's what the plant would normally take. It's not receiving any of these other fancy treatments. It's not getting the pop, it's not getting the juice, it's getting the water. So that's something that's pretty normal, and we're not going to change what we normally give the plant. So that makes it a control group. So more about control groups. Like I said, it's the group in an experiment that's not changed, and we use that for comparison purposes. So because we're not changing the water, we're able to see exactly how juice affects the plant and exactly how pop affects the plant. It's useful to have control groups because then we can compare the response of the experimental groups. When we do that, we increase the validity of our experiment and the relationship between the experimental group and the things that we're testing. Now, the experimental group is the group that gets the treatment of the manipulated variable. So in the example we just saw, the experimental group would be the group that's getting a different liquid that's not water, so the pop or the juice. But not all experiments have a control group, and that's okay. But a control group is a really, really good thing because it increases the validity of our experiment. We can compare that to the experimental group. Here's another example. So the experimental group will be the group that gets sprayed with this biofertilizer X. The control group is not going to get sprayed with this biofertilizer X. That way we can see the effect that this biofertilizer has on the plant growth versus if it's not getting that, what happens? A good control group should be kind of identical to the experimental group in all ways, except for the difference is that you're not getting the biofertilizer X. So how can scientific research be communicated? There's two ways that we can communicate scientific research. The first one is quantitatively. That means numerical data or numbers. We can compare that, or we can communicate that with graphs, tables, charts, all types of stuff. So when we look at this picture, we see 13 trees. That's a quantitative number. That's quantitative data. The other way is qualitatively, and that's descriptive data, like pictures and words. So when we look at this, we would say yellow, blue, and red birds. So it's pictures, it's word descriptions. Oftentimes it's diagrams. Communication is really important in how we communicate our scientific research because it allows for ideas and results to be shared and new ideas to be generated. Oftentimes, scientists use both of these to communicate their findings because they make it more valid. To have both means that it worked out both ways. You could see numerically and you can see with word descriptions and pictures and diagrams that they both worked out. Okay, last slide for this. Parts of a graph. So I'm sure you've seen graphs in math class, and graphs are really, really important in science. Graphs are one of the main ways that scientists communicate those relationships that they found. So the big part, you need to have a title on a graph. You need to have your x-axis as your manipulated variable or your independent variable. The y-axis will be your responding variable or your dependent variable, The scale, which is on your axis, should be evenly spaced. It shouldn't go 2, 10, 12, 18, 22, 30, 100. That doesn't make sense. It needs to be evenly spaced. We go 2, 4, 6, 8, 10, 12, 14, 16. The big part is that we start at 0. We can't have this number down here be like a billion. It just doesn't show the real relationship. We need to start the graph at 0. So that concludes this note video, and I will see you guys when I see you.